looking now at colorectal cancer liver metastasis, which was an interest originally developed in Peter's lab by Sébastien. I'm not sure if there was somebody else before Sébastien, but obviously he's taken this on and has done real interesting work. Sébastien, as the name implies, is from France. He did his DESS at the Université Aix-Marseille in France, and then decided to hop the big lake and come to Quebec City to do his PhD with at the Université Laval with Lucie Janot working on HOX5 gene regulation, and then move down to Montreal to do a postdoctoral fellowship with Peter Siegel, where he was actually first looking at breast cancer metastasis to the liver, and has stayed on as a research associate with Peter, but focusing mainly on colorectal and breast, both of them, I guess, Sébastien? Metastasis. So we're happy that you could join us and that you can show us what happens in the case of colorectal.
Yeah, so thanks for giving me the opportunity to present our PDX project about colorectal cancer. Thanks, Peter, for switching the slides. So as Peter briefly mentioned, and similarly to what he describes about breast brain metastasis, colorectal liver metastasis also presents with different growth patterns. There's mainly 3 of them that have been described in 2001, initially in patient specimens. So I will focus mostly on 2 of those growth patterns for my talk, because the pushing type is quite rare and we won't focus on it. So one of the 2 growth patterns we are interested in is the desmoplastic growth pattern, in which the cancer cells are insulated from the parenchyma by a desmoplastic ring. So there's no direct contact between the cancer cells and the hepatocytes. And these tumors are really characterized by high androgenic characteristics. On the other hand, we do have the replacement type lesions, in which the lesions are growing by replacing existing hepatocytes, and they are just co-opting the existing vasculature of the liver. So the reason why we've just started our PDX program for CRC liver metastasis is just because if you're using cell line models, there's mostly only giving rise to replacement type lesions using experimental assays. So there was no existing tools to study those desmoplastic type lesions, and it was really a challenge for us to see if we would be able to establish such type of lesions. So why we are so interested in between those desmoplastic versus replacement type lesion is just mostly because in the patient there's clearly different clinical outcomes. With the patients with the desmoplastic type lesions that are behaving a lot better than the one with non-desmoplastic type lesions. And that's been confirmed in more than 2,000 patients now, that the desmoplastic lesions behave a lot better. And similar observation has been also made in liver metastasis from other type of cancers that will form the different growth patterns, such as the uveal melanoma, the skin melanomas, or even the breast carcinomas. So the reason why those desmoplastic lesions behave better is just because they are most likely to respond to anti-angiogenic therapies. On the other hand, the replacement type lesions really behave the worst, and they're not responsive to any anti-angiogenic treatments. However, it seems that they somehow respond a bit to chemotherapy, as you can see on the right of the slide. So following chemotherapy treatments, the patients with the replacement type growth patterns is decreased from 81% to 70%, suggesting that if they would respond to a treatment, that's the only way for now just to treat those patients. Yeah, thanks. So this is the pipeline for just PDX program for CRC liver metastasis. So as Peter already mentioned, there's a need to set up a couple of collaborations, especially with surgeons. So for our program, we are just collaborating with the group of Dr. Peter Metrakos, which is an hepatobiliary HPV, hepatobiliary pancreatic cancer surgeon at the MUHC. So we just get the surgical material from the OR, and this is for sure, as Peter mentioned, patient material is biobanked, either as OCT material, paraffin, flash frozen, and also banked as PCs to further reestablish the PDXs if needed. And some material is freshly xenographed into for CRC PDXs, it's only works better in SCID-beige background rather than any other mouse strains. So we either establish them as a subcutaneous lesions or directly through intrahepatic transplantations. So can we just go one back? So whenever they took as intrahepatic or subcutaneous tumors, there will be a passage at least 3 times and material banked all the time. Okay. Next slide, please. Okay, so doing so, we've been really successful in establishing both types of lesions. So replacement type lesions from the donor, being able to recapitulate the growth patterns whenever they've been implanted intrahepatically. And for the first time, to our knowledge, we've been also successfully able to recapitulate desmoplastic growth patterns in the liver following intrahepatic implantation of desmoplastic type lesions from the donor. So far, we've been able to establish up to 11 PDXs from replacement type donor lesions with a take rate of about 85%. Our take rate following intrahepatic transplantation is about 70%. Most importantly, can you just one back, please? All the ones that were successfully implanted intrahepatically recapitulates the growth pattern seen in the donor. So all the lesions were giving rise to replacement type lesions. Next slide. Please. Thank you. About the desmoplastic type lesions, we've been able to successfully establish 7 of them with a take rate of 64%. So going back, we assume that the desmoplastic type lesions are less aggressive, and as you can see, the take rate seems to be a bit lower. It just tracks with what we've already discussed before. Similarly, whenever they are established in the liver, they just recapitulate the growth patterns seen in the from the donor. Beside these fully replacements or fully desmoplastic type lesions, we also had a chance to access a couple of donors with mixed growth patterns. And we've been successfully able to establish 5 of them with a take rate of 71%. And what's really interesting, it's in that case, whenever the donor is mixed, it can give rise to either one type of growth patterns or the other when transplanted into the liver. So, and it's a bit, can you one slide back? It's a bit more challenging, but we've also been successful in getting PDXs out of biopsy, which has a lot less material, but we've been with a 60% take rate, been able to succeed in establishing some. But we don't know what the growth patterns of the piece that we've been given was to get started with. So next slide. So now we wanted to use those PDXs to study our mediators of liver metastasis. So we've initially notified in the lab Claudine-2 as a functional mediator of breast cancer liver metastasis. And what's really important is that breast cancer metastasis will only be most likely of the replacement type. So over 95% of liver metastasis from breast cancer will be of the replacement type. So we next wondered if Claudine-2 would be a specific mediator of the replacement type growth patterns in CRC. So in collaboration with Peter Metrakos at the Glen, we've did RNA sequencing of a subset of samples either from desmoplastic type lesions, or from replacement type lesions. So material was collected either from the core of the lesions or from the adjacent tissue. And this was done on 9 desmoplastic lab lesions versus 6 replacement type lesions, versus 9 desmoplastic type lesions. And interestingly enough, all the tumor samples, regardless of the growth patterns, were clustering together, and all the normal tissues clustering together. And we've first did a targeted study. So we just asked about the expression profile of the Claudin family members in those samples. And doing so, we've been able to identify four groups of expression of patterns of expression of Claudins. So some Claudins that will be found expressed at high levels in both the desmoplastic of the replacement type, including Claudin 4, 7, and 3. Some Claudins that were just expressed highly in the replacement tumors and the normal, including the subset at the bottom. And the one we were most interested in. So some Claudins that were found specifically expressed in the replacement type growth patterns. That would include Claudin 2, as we would have expected from our initial findings in breast. And we were also happy to see that Claudin 8 was specifically expressed in the desmoplastic type lesions. So we next decided, we'll go to the next slide, Peter. So we next wanted to validate those RNA-seq data at the protein level, doing immunohistochemistry on liver metastasis from patients. And we first validated from patients obtained from our collaborators at the McGill MUHC from Dr. Petrakos. That would include the patients that have been RNA-seqed. And we've been able to validate that Claudin 4 was highly expressed in both growth patterns, that Claudin 8 was enriched in the desmoplastic type lesions, while the replacement type lesions were characterized with an increased Claudin 2 expression levels. Next, in collaboration with a group in Europe, we've been able to access more samples as a validation cohort. And once again, we found that Claudin 4 was highly expressed in both types of liver metastasis. There was still a decrease in Claudin 8 expression in the replacement type lesions, and an increase in Claudin 2 expression in those replacement type lesions in both cohorts. So, and then we wanted to assess if our PDX's models were recapitulating that Claudin expression patterns seen in the patients from the RNA-seq data. And we were happy to see that Claudin 4, as expected from the previous data, was similarly expressed in both growth pattern. Claudin 8, even though it doesn't reach statistical significance, there was a trend for a reduction in Claudin 8 expression at the replacement type lesions. And really importantly, we found that Claudin 2 was enriched in the replacement type lesions compared to the desmoplastic type lesions. Suggesting that our PDXs, at least considering Claudin expressions, recapitulates what's seen in the patients. So the usual way just to study the role of a gene in metastasis, especially in liver metastasis, would be just to manipulate its expression in a relevant cell line model. So for the case of colorectal cancer, it would have been HT29 cell lines. Unfortunately, as I mentioned earlier, all cell line models will just give rise to only replacement type lesions. None of them, to our knowledge, is able to recapitulate the desmoplastic type lesions, so which makes it really difficult just to use such models. Considering that Claudin 2 seems to be involved in the replacement type lesions, we did that exercise. So if we knock out its expression in the HT29, we see a reduction in their ability to form liver metastasis. And this effect was really mediated through Claudin 2, as its re-expression just reskews the phenotype. And interestingly enough, also it's also true whenever we do an orthotopic injection of those HT29 cells following cecal injections. So the Claudin 2 knockout mice don't form any liver metastasis following intracecal injections compared to the parental cell lines. While there was no effect on the primary tumor growth. So it's really the metastatic potential which is affected. So how could we then study those desmoplastic type lesions if no tools is available, no cell lines available to recapitulate those that pattern? So we thought that it would be one way would be just to establish organoids out of our PDXs. And this work has been done in collaboration with Dr. Grigoriev at the MUHC. So organoids been derived either from the desmoplastic type PDXs or from the replacement type PDXs. And what we found is that following re-injection of these organoids in the liver, they were still able to recapitulate the growth patterns from the donors. So which was really a really important aspect for us. So suggesting that we've been able to put in place a system that could be manipulated eventually. We next, can I get to the next slide? So we next wonder if those organoids would have retained the claudin expression patterns seen in the PDXs and in the patients. And it was indeed the case. So replacement type derived organoids were expressing claudin 2 while the desmoplastic one were characterized by a claudin 8 expressing pattern. Claudin 4 was expressed in all of them, regardless of the growth pattern. So the idea beside the establishment of the organoids is just being able to manipulate them. So the first experiments we did was just to see if we can just infect them with a luciferase reporter in order just to be able to track them over time for further experiments for treatment. Then we can assess at what time to get started the treatments and so on. So we've been successfully able just to either manipulate desmoplastic type lesion or replacement type lesions to make them being able to express the luciferase. And following that manipulation, they still retain their ability just to grow as the expected growth patterns. So either replacement or desmoplastic, which was really great. So, and this tools helps us to monitor tumor growth over time, which is really great. So we are also been able to manipulate expression levels of claudin 2 in some of these PDXs, and the in vivo experiments are ongoing. So I hope they will retain their ability. And the idea will be to eventually switch from one pattern to the other. So as a summary, we've established about 30 PDXs models of colorectal liver metastasis. Out of those 30, I should mention that 4 of them are matched sets of either primary tumors, derived PDXs, and liver met PDXs from the same patients, because surgeries of the primary tumors and the liver metastasis been done the same day. We also have a set of 2 patients from which we had established PDXs from recurrent liver metastasis. So one case, we had the primary tumor and 4 recurrent liver metastasis, and the other one is just 1 recurrence of the liver metastasis. So we've been able to show you, I hope, that the PDXs models that we've established were able to recapitulate the 2 growth patterns if we're focusing on the desmoplastic versus the replacement. At least considering the claudin expression patterns, both the PDXs, for the PDXs were just exhibiting a high degree of concordance with what was observed in the clinical material. And we think that really in the near future, those PDXs models will represent an extremely valuable tool just to test functional contribution of potential mediators, especially if we're considering of claudin 2. What if we re-express claudin 2 in a desmoplastic type organoids, would it then lead to the formation of replacement type growth lesions? And I think also that those models could represent excellent models for preclinical therapy trials. And we are currently trying to assess the potential, the therapeutic of antibodies against claudin 2 for the treatment of those patients with specifically the replacement type lesions. So,
thank you, Sébastien. Are there questions on Sébastien's presentation? Other than the one I'm about to ask him. I should remember this, but I don't. Do desmoplastic tumors ever evolve to become replacement? I don't think so, but I'm not sure.
They do. Okay, so if what's been observed that whenever you treat desmoplastic type lesion, most of the time, whenever they recur, they have a tendency to recur more often as replacement type lesions. So
we know do we know the change in genetic program that occurs then?
no, not to my knowledge.
Not
yet.
Exactly. Okay.
So the question would be, were there replacement type cells within the original desmal lesion that just grew out, or is there a possibility to convert, right? And I think that's an open question.
Are there any questions? If not, then